Hello, everybody. Today we're going to talk about DNA replication in the cell cycle. It's an important aspect of cell biology and biochemistry, has incredibly important implications for cancer therapy and treatment. So let's take a look. So the cell cycle starts with G1. Basically, all cells are born in G1. Here you see a cell with two chromosomes. Basically, it's diploid in a sense, so we're ignoring all the other chromosomes that you'd find in a human cell for simplicity. And so in G1, it's a period where the cells are growing. And what they do is they're actively assessing how much nutrients they have, and they have to make this decision in order to enter S phase. And what happens is that in yeast, there's a phase called start, or it's the restriction point in mammalian cells. This is the point where growth factors will enter and then actually signal for the cell to go from G1 into S phase. And S phase is a big deal because you need a lot of energy and you need a lot of material to actually synthesize in humans 3 × 10⁹ base pairs, times all the number of chromosomes you have. And so in S phase, what happens is that you actually duplicate the chromosomes as you see here. And what we can see now is that we have a centromere with 2 attached sister chromatids. And then after S phase, the G2 phase will survey and make sure that everything was faithfully replicated during S phase, and then the cell prepares itself for another dramatic event, and that is mitosis. And during mitosis, you'll actually see condensation of the chromosomes. They'll become much more clear under a microscope and visible. And then what happens is that ultimately what you do is you segregate the replicated chromosomes into daughter cells, and now you are born in G1. Now, there's also G0, and this is during a quiescent phase, where if there's not enough nutrients, the cells can actually exit the cell cycle, G0 phase. So the phases of the cell cycle are governed by the action of cyclin-dependent kinases, also known as CDKs. And as you might expect, they phosphorylate a number of targets, and these targets that are phosphorylated in each of the phases can either activate or inactivate a number of signaling pathways that ultimately lead to the definition of the cell cycle phase itself. So what we see is that CDK cyclin D is indicative of the G1 phase. And as CDK phosphorylates substrates, what you do is you progress through G1 phase, and then the decision to enter S phase is given by cyclin E, and cyclin E promotes S phase, and then the G2M transition is governed by cyclin B. There's actually a lot of other cyclins. We're just looking at a very simplistic view of it. And as the name might imply, cyclins, the concentration of cyclins fluctuates as a function of the cell cycle. So we see cyclin D builds up where its maximal levels are in G1 and S phase. And as you get into mitosis, it ultimately is degraded. Cyclin E is built up right before S phase, and then it disappears right after S phase. And with CDK1, also known as cyclin B, what we see are maximum levels of mitosis, then it precipitously falls off in anaphase. So here are the stages of mitosis, and what you see is a cell in G2. We see the nucleus. We don't see a really good definition of the chromosomes yet. They haven't constricted like they do in N phase. And what we see are the centrosomes. Centrosomes are duplicated in animal cells in S phase. And here what we see are microtubule asters that are going to ultimately organize and position in spatially position the actual spindle. And so the centrosomes are going to migrate to opposite sides, and they form these microtubule spindles. And then what happens is that as we're entering M phase, cyclin B is going to start phosphorylating a number of targets. One target actually causes the breakdown of the nuclear envelope. It's the phosphorylation of lamins. And what you see here is a disintegration of the nuclear envelope in prometaphase. And then the actual chromosomes will line up on the metaphase plate here. And then what happens during anaphase is that you segregate them to the opposite poles. And then telophase will introduce a cleavage furrow. And then ultimately what you do is you separate. Now, in many cases, what happens is that, well, actually, it's probably rare, but there's certainly cases by which you don't properly separate chromosomes in anaphase, and this leads to a process, at least in meiosis, known as non-disjunction. You can get a number of developmental abnormalities as a result. So how do cells actually go into S phase and conduct DNA replication? So in the early 60s, Francois Jacob, he's the guy who discovered messenger RNA. He dreamt up the replicator hypothesis. And in E. coli, which has one origin of replication called OREC, he postulated that a protein, which we now know as DNA, would act in trans, and it would diffuse to a cis-acting element that would bind it. And what that would do is serve to nucleate a site by which replication polymerases would associate. And then what we see here is that the origin recruits the polymerases in blue, and these are the actual primers. These RNA primers are used to recruit DNA polymerases, and you would generate the canonical DNA replication bubble, which would serve to propagate two forks in opposite directions. Now, a long time ago, Leonard Hayflick recognized that fibroblast cells in culture had a finite replicative capacity. That is, he can grow them for a period of time. So here you see an exponential phase of growth, and then what happened, they'd maximize, and then it would senesce, they'd stop growing. And what he came up with is the idea of senescence, that this is called the Hayflick limit. But what we know is that cancer cells actually go through a process known as crisis. They don't have a Hayflick limit. So there's a number of things happening at this particular junction here where they choose not to senesce, but actually generate what we call as an immortal lifespan. And there's a number of things going on at the cellular and molecular level that people have been deciphering for many years with respect to cancer. And one of them is with respect to telomeres. And we know that telomeres are the ends of chromosomes. And so you can think of like them being like, you know, the ends of shoestring have that plastic around it that actually keeps the ends from being frayed. And if you look at the ends of linear chromosomes, so this would exclude bacterial chromosomes because they're circularized, what you would find is these repeating segments. Okay, this is a human telomere. Other types of animals like mice have different repeating segments. But what you notice that there's a single stranded DNA component such that the 3' end is an overhang. There's actually no double-stranded component of it. And what this does, it creates a big problem known as the end replication problem. And why is that a problem? Because what you need in order to synthesize new DNA by DNA polymerase is it extends, as you see with the arrow, in the 3' direction because it's a 3' hydroxyl extension. We'll look at this in more detail in a moment, but there's actually no template. How does it know what to put down? There's nothing for it to do. But what the cells have engineered is an enzyme called telomerase, which actually has an RNA catalytic component by which this RNA sequence here actually serves as the template to extend the actual telomere end. And so the end replication problem is solved by the action of telomerase. So it turns out that the reason why cells senesce is because what happens over time is that their telomeres shorten. And the reason why they shorten is because many of our cells don't actually express telomerase. It's only our germ cells that primarily express it. And as the telomeres shorten over time, they're recognized as a DNA double-stranded break, which you're going to see a lot of in a few minutes. And what happens is that the cells see that DNA break as they perceive the reduced telomere size, and they stop their growth and senesce. So telomere shortening is believed to explain the actual Hayflick limit. And what happens then is that cancer cells are going to reactivate their telomerase. And so you can imagine that telomerase is a major cancer therapy line of research. So we go to DNA replication, we look at how this all started. And we know that Watson and Crick published a paper in 1953 in Nature showing the actual structure of DNA. They used Chargaff's base pair ratios as well as a number of structural experiments. This is all published, you know, but also there's a great book called The Double Helix. I would highly recommend reading this. It's incredibly fun. So after the structure of DNA was published, the issue of how it replicated became the chief concern of a number of researchers. And so Meselson and Stahl did an experiment known as the Meselson-Stahl experiment that proved that DNA replication was semi-conservative. And what they did was they actually grew a culture of E. coli in N14 nitrogen, as represented by red, and another culture in parallel in N15. And what they can do is actually sediment the genome after it replicated in each of the isotopes. And what you would see is that they would separate in the gradient at different layers because of the nature of one's heavier than the other. But so then the experiment was to grow cells in N15 media and then transfer to N14 media and monitor the cell divisions. And so what they came up with was that they observed that what you start out with in N15, and then as you go to the N14, you get an intermediate here, and then ultimately you can separate into two. And then as you move on, the N15 disappears. And what they came up with was that what was happening is that each strand of the DNA double helix would serve as a template for polymerases. And so what they had was an N15, N14 model. This is the semi-conservative replication model of Meselson and Stahl. So shortly afterwards, what we saw was that Roger Kornberg, as well as his father, were purifying DNA polymerases. And what we see here is the classical fork model. This is Kornberg's purification of RNA polymerase. So what we have is the leading strand and the lagging strand model. So we know, as Kornberg discovered, that all DNA polymerases use RNA primers to actually recruit the polymerase to a specific spot. We know that the cells produce it by an enzyme known as primase. And so the leading strand goes in 5' to 3' direction. And then the lagging strand has to open up first, and then you layer it down in what we know as Okazaki fragments. There's a helicase ahead of it that's unwinding the DNA double helix. But as you unwind it, what happens is that you can think of like a rubber band getting all twisted up. You have to actually relieve the torsional stress, and that's what topoisomerases do. Actually, there's a lot of other proteins that are acting on this particular spot, but I think that for simplicity, this is the best way to start. So what happens then is that the DNA polymerase is going to use a 3' hydroxyl group as a nucleophile to actually attack in an SN2 fashion the alpha phosphate of an incoming DNTP base. The base that comes in is by definition complementary to its template strand, which is not shown here, and you release inorganic pyrophosphate, which will be hydrolyzed as a way to drive the reaction forward thermodynamically. So people had come up with chain terminators. We saw a little bit of this before when we talked about viruses. And so we talked about AZT is if you're missing a hydroxyl group, what happens is that you chain terminate. Or here's a dideoxy. AZT is an HIV drug, or it's a first generation HIV drug, probably not used too much anymore. And so what happens is that you can actually terminate the polymerase by taking away its 3' hydroxyl nucleophilic group. And so Fred Sanger in the 70s recognized this, and what he did to win his second Nobel Prize was he created dideoxys, and it turned out that probably synthesizing these things in the lab was the hardest part. So this is the dideoxy Sanger sequencing technique. It's actually listed on the AAMC MCAT content outline. And so here's what you do. You take a template DNA and you add enzyme, the primer, the buffer, and you add all 4 of the deoxynucleotide triphosphates, GATC. You mix it up, and then you aliquot it into 4 separate tubes, where each tube contains a dideoxy G, A, T, and C. And what will happen is that you will incorporate the normal DNTP in some of the samples, but as you incorporate the dideoxy, you'll chain terminate such that you'll generate what's known as a nested set of fragments. So if you look at the top 2, the green ones here, which is G, in the top one, what you see is that both G or all three Gs got incorporated, but the second one, only 2 of them did. And then as the dideoxy enters, you stop polymerizing and you synthesize a DNA strand that has 1 less base pair than the one before it. And what you can do is you can actually run all these reactions out on an acrylamide gel, which will separate as a function of the size of the synthesized DNA chain. And what you can do then is actually read the DNA sequence, and you read from top to bottom. In this case, it goes CC, GATC, TAGG is the actual DNA sequence. So the Sanger dideoxy technique is important. It's how we sequence the genome. So DNA structure and mutation are intrinsically linked to each other. And it's based upon the chemistry. First of all, the polymerase makes mistakes. People say that in eukaryotes, there's like 1 mistake every 10 to the 7 or 10 million. But if you think about the fact that you have 3 billion base pairs, what is that, like 300 mutations on average per cell division cycle? But what if your cells divide multiple times over a week or a month? So you can start introducing a lot of mutations. We have a repair apparatus, a DNA mismatch apparatus that's designed to actually weed out these errors, but in itself may not be 100% efficient. But the actual chemistry of the base pairs themselves creates the idea of mutations. And this is something that Watson and Crick struggled with. And this gets to the notion of tautomers. And this is also on the AAMC MCAT outline. So we're going to look at more of an organic chemistry argument for this. So if you look at the thymine, for example, T, it exists in a keto and an enol form. And there's the lactam and the lactim. You should appreciate that a lactam is basically a cyclical amide. Okay, here's a C double bond O and the N as well. And over here, it can also exist in what's known as a tautomer, which is a constitutional isomer, as an enol. And so how does this actually work? Well, I'll show you in a minute, but in most cases, the keto is 99% of the time it's in the keto form. So the equilibrium's heavily favored for the keto. There are some exceptions to this. For example, the molecule phenol is mostly in the enol form, and that's because of resonance. And there's also some hydrogen bonding effects that can stabilize keto forms. But the vast majority of the time, it's going to be in the keto form. And so the mechanism of this is that you have this acidic proton right here, and a base that's from the solution will pull it off, and these electrons will come in and occupy this, and you generate a negative charge on the nitrogen. This, by resonance, pushes up the electrons here, and what you get is an enolate. And then if you have a proton come into this from the solution, you can generate an enol. And this goes back and forth, back and forth. But what it has implications for with respect to mutations is that a base like thymine, 1% of the time can exist in the enol form. And what you recognize is that in the keto form up here, the C double bond O, this is a hydrogen bond acceptor. But in the enol form, it's a hydrogen bond donor. Okay, so as a consequence of that, when you generate a new hydrogen bond donor in thymine, by virtue of it being in the enol form, what you can do is have an abnormal base pairing event where the T can bind to the G by hydrogen bonding. So this is how you could actually insert the DNA polymerase can come by at that one moment in time where the enol form is favored and insert the wrong base pair there. So tautomers and mutations are intrinsically linked through the polymerase replication apparatus, and the fundamental principles are built through organic chemistry. So there's even more about DNA replication forks. So what they can also do is create DNA double-stranded breaks, and we're going to see a really good passage on the BRCA genes today about that. So here's what we have. We have a replication fork, as we saw before, but this time there's a nick in the DNA. It's like a pothole, right? And so how do these form? Well, they form all the time. There's thousands and thousands of them being formed every second. So what happens then is that free radicals can unseal phosphodiester bonds. And in many circumstances, as I'll show you, you're going to fix that nick, you're going to fix the pothole before the polymerase drives over it. But in the cases where you don't do that, as you can see, if the polymerase arrives before the nick is fixed, what do you have? A DNA double-stranded break over here. And so imagine that that occurs, and then we have to fix it. And the cell knows this is going to happen all the time. So how does it fix it? Well, the way it fixes a DNA double-strand break in S and G2 phase is through a process known as homologous recombination, as you see here. We call it HR. It occurs in S and G2. So what happens, we make our break, and then the cells have nucleases that are going to chop back to create these 3' overhangs here. And this 3' overhang has a hydroxyl group. And since we're in S phase, hopefully we have our sister chromatin right next door to us. And so what you can do is have this magical event that's conducted by RAD51, BRCA, and a lot of other factors that we'll become acquainted with. What they can do is invade the homologous sequence. And what you notice as you form a D loop or a holiday structure, if you look on the side, it looks like a D. And what happens then is this 3' hydroxyl group is basically creating a new replication fork, showing us that homologous recombination and DNA replication and DNA repair are all kind of the same thing. But what if the DNA break happened in G1 phase? It didn't happen by the polymerase finding a nick, but rather it happened because you got exposed to an X-ray or something like that, ionizing radiation. You don't have a sister chromatid to rely on for this process. So the cells do something called non-homologous end joining, or NHEJ, excuse me. So we have DNA damage, creates the breaks right here. And then what happens is that the cells kind of just stick the two ends together. They don't really care what was missing, which could be a problem in some cases. But given the fact that a lot of the genome has what they call junk DNA, or it doesn't really code for a lot, it may not be such a bad idea after all. But you know, what's going to happen is that as you're trying to fix and repair these breaks in the G1 phase, you don't want the cell cycle to keep going. That's a super bad idea to enter S phase with broken DNA. And so what happens is that this DNA damage is sensed by ATM. It phosphorylates a famous protein known as P53. And P53 can actually make a protein called P21. I kind of think we should be in the mood to understand and know these names. These are famous name proteins. And what happens then is that P21 inhibits Cyclin E, and it prevents the actual entry into S phase. It also probably inhibits Cyclin D. So P53 is a tumor suppressor because it's suppressing the ability of the cell to perform the cell cycle under the context of DNA damage. So here's the summary of fixing the DNA breaks. It depends upon what phase of the cell cycle the break happens. In G1, you do non-homologous end joining. You may miss out on an intervening piece of DNA sequence. And those of you who've had immunology, and we have a workshop on immunology because it's on the MCAT, probably recognize that the way you generate antibody diversity is through non-homologous end joining. And in the case of homologous recombination, or HR, I've now added RAD51 and BRCA as these proteins that endow this single strand with the ability to invade its homologous sequence in order to conduct replication and repair. So one of the important things about translocations is that when we break our DNA, what happens is that not in all cases will you just magically fix it by end joining or homologous recombination. So what can happen is that when you have these DNA breaks, these ends are promiscuous, and in some cases they cause a lot of problems. They cause translocations. And this case is, I'm showing you now, is called Burkitt lymphoma. And in Burkitt lymphoma, what you see is that immunoglobulin H locus and the c-MYC locus, actually there's the breaks in immunoglobulin H, and it invades the c-MYC chromosome such that you actually swap out and you put the c-MYC gene under the IGH promoter and the cell starts cranking out c-MYC. Very bad idea, because c-MYC regulates about 30% of the gene, and so the chromosome, excuse me, and what you're doing is you're going to promote oncogenesis, rapid uncontrolled cell division. There's also BCR-ABL. For example, chromosome 9 and 22 have this weird affinity with each other, so you can create breaks at these loci here. And it turns out by translocation, you juxtapose these two together. So normally BCR is a protein that would never even hook up with ABL, but under a condition where you have DNA damage and breaks and you don't fix them properly, what you can do is you can juxtapose BCR and ABL together, and you magically make a new protein. This chimeric Frankenstein protein actually encodes a tyrosine kinase, taking over cells, and you get chronic myeloid leukemia, CML. So guys that come up with tyrosine kinase inhibitors that are designed to treat this condition, this specific chromosome is known as the Philadelphia chromosome. So we're going to take a look at a couple of workshop passages that speak to understanding cell cycle translocations. We're going to combine biochemistry, genetics, and cell biology. And so I hope that you guys got something out of that, and we'd like to see you later.